It's a pleasure to be here. Last time I was here was 20 years ago, so I had no gray hair. I had more hair, less pounds on. I was telling Tom that, but the place, this hall didn't exist, but the place, many parts look the same. Richland definitely does not look the same. It's changed a lot. So it's really my pleasure being here. What I'm going to try to tell you today is the ways that we use machine learning in metabolomics and how some of the new types of data that we're generating and really the challenges that we encounter almost on a daily basis when it comes to machine learning and metabolomics. So yeah, there we go. So I'm sure that many of you know quite a bit about metabolomics. But for those of you that don't know about metabolomics, it's basically the field of omics that deals with small molecules, particularly metabolites. They can be primary, secondary metabolites, also deals with xenobiotics. In general, with small molecules, and we distinguish ourselves from proteomics based on that kind of like operational difference on the molecular weight range of molecules that we look at. I don't know if you guys have seen the show, The Dropout, but if you haven't, you should. But The Dropout is a show about a startup company that was interested in doing diagnostics with a single drop of blood. And it was a big disaster because they were promising things that really couldn't be done at that point. But that doesn't mean that it won't be able to do that. So I think that we will, probably in 10, 20 years, you go to the doctor and rather than getting a cholesterol test, you're going to get a metabolome test that will give the doctor many more reasons to tell you why you need to take Lipitor, basically. But it's going to be a much more powerful data set that comes from that single analysis. In metabolomics, the simplest experiment that you're going to see in metabolomics is a case control experiment where you're looking at, let's say the differences between healthy individuals and individuals with a certain disease, or you could see treatment 1 versus treatment 2. And typically real life it's never that simple. So I'm going to show you some case studies where you're going to realize that things get complicated very quickly. But sometimes it's and Olga was talking about the mixed linear model, how you can decompose different sources of variance. That is becomes quite complicated quite quickly. So many of you are, I'm sure, are experts in machine learning. I am not. But I realized that our research group has gone from being, a chemistry, mass spectrometry research group to being a group that does a lot of machine learning. And I think that that's a reflection of where the field is going, where non-experts are doing machine learning, and it's becoming a very powerful tool that can also be misused if you don't use it properly. So these are the classic things that your tasks that you do in machine learning, typically supervised or unsupervised learning. And pretty much all of these play a role in metabolomics. So I'll show you examples based on different applications. We've been very interested in ovarian cancer, partially because Atlanta, where Georgia Tech is, has several large hospitals that receive very large numbers of patients with ovarian cancer. Ovarian cancer is actually a relatively rare cancer, but considering that it's quite rare, the mortality is quite high. And that's because it's very difficult to detect. When you go to the OBGYN and there's no simple test or simple screening test that you can catch it at an early stage disease. Therefore, you see here that it's the 5th leading cause of death, which is, it shouldn't be, basically. If you catch it early, like most cancers, the survival rate is very high, a 5-year survival rate of about 29%. But if you don't, which is many times what happens, then it's not so survivable. There's no obvious techniques. There's no obvious biomarkers of ovarian cancer. Actually, there's some pretty famous papers on ovarian cancer that were very wrong. They were, that are used as examples of how not to use machine learning for do discovery, biomarker discovery. But one of the biomarkers that is still around is called CA125. It's a mucin protein, and per se, it doesn't have a lot of predictive value. But if you look at the trajectory over time of CA125, it can actually have some value, mostly in high-risk patients, patients that have BRCA mutations, for example. But still, there's a big need in terms of diagnostics and detection. So I'm sorry, I didn't warn you, this is right after breakfast. Hopefully nobody's queasy, but it's difficult to study rare diseases in humans because it's rare to find the cases to study them. So one of the things that we do is do quite a bit of work with animal models of ovarian cancer. These are the two of the most successful animal models. And the reason why I'm showing you these pictures here is because I'm going to tell you about some metabolomics results in a second, but we work with a double knockout mouse model that has mutations in two cancer-related genes called DICER and PTEN. And what happens is this is the reproductive system of a mouse, and this here and that there, those are the ovaries. And one thing that you may not know is that ovarian cancer is actually not ovarian cancer. Ovarian cancer actually starts in the fallopian tubes. So it should be called fallopian tube cancer, and then it migrates to the ovaries. So what you see here is massive tumors, in this case in both ovaries, and these are the ovaries themselves. The fallopian tubes are behind here, you cannot really see them. And this is the equivalent of what in a human would be the uterus. It's not the same in a mouse. And this, of course, it's a very late stage ovarian cancer. This is when it's metastasized. This is a triple knockout model where, in addition to the previous two mutations, it has a p53 mutation, which is a mutation that in humans occurs very often when ovarian cancer patients. I am not an expert, although we've been working on this for almost 10 years now, but my colleague Young Kim at Indiana and Martin Matsuk at Baylor are really the world experts in developing these mouse models. And what's really interesting is that they really mimic the disease in humans. So one of the things that we've been doing is we've been trying to basically establish what does it mean really early stage ovarian cancer. And that's a little bit of a subjective, classification because it's a continuum of disease. But we've been, with these animal models, you can actually do longitudinal studies throughout the lifetime of the animals. And I'm going to show you some results, for example, for the DKO animal model. What we do is we start sampling blood from the tail of the animals, from the moment that they're old enough to basically be sampled. It's not an easy thing to do. And then we measure every 2 weeks all the way until the animal has to be euthanized because the cancer is too advanced. And we apply a pretty robust lipidomics platform. We do lipidomics by liquid chromatography mass spectrometry and tandem mass spectrometry. Typically, we use Orbitrap mass spectrometers for this. They're very robust and very stable. And then we generate quite complex data sets, very robust data sets. And something that you may be interested in is that these data sets are published. We publish them because we cannot expect to be able to analyze all the data in our lifetime, all the lifetime of a grad student in grad school. So I'll just show you a little bit of the different things that we do in terms of machine learning. So the raw data is very rich. This is what the raw data looks like when it comes out of the instrument. This is the retention time in the LC column. This is the abundance of the different ions. These are not pure peaks. There's many species. There's thousands of species. And this is what we do, different types of chromatography. So we do chromatography for the lipids in both positive ionization and negative ionization. And we also do chromatography for the polar species, which sometimes contains interesting information, just to get the most coverage that you can expect. And then to identify the species, one of the things that we do is we do tandem mass spectrometry. So we look at the pieces of the molecules and we try to identify. The keyword being try, because it's not always successful. So just to, sorry, it took a little bit to load. One of the things that you see in this type of data sets very quickly is that many of the molecules detected follow similar trends over time. So you can apply clustering techniques to these, for example, these lipid trajectories, we call them. And you see here, for example, this cluster is an interesting one. These are a cluster of molecules, in particularly lipids, that they all have the similar trend over time. 1, 2, 3, 4, 5. Those are the different lifetime stages that we define. But of course, we are particularly interested in lifetime stage 2 because this is what would allow us to do early detection, right? This is an early enough stage. So then you can ask yourselves, well, what molecules are these? and what are they contributing to. And we typically focus first on molecules that we have some degree of annotation, and which is roughly 10 to 20% of the data set. So it's a small fraction. And these heat maps here show you the names of the lipids and their abundances for different stages. And notice that the names are quite complicated. That's because you can annotate lipids with different levels of detail, depending on the information, the spectral information that you're obtaining in that specific case. So this is particular notation that is used to annotate lipids. But you know, we can group this by families. And for example, in this cluster B that I mentioned to you, most of them were from the phosphatidylcholine family, which are membrane lipids. And of course, cancer is associated with cell growth and therefore a lot of membrane remodeling. But you know, this is the type of things that you can do on a first pass. Of course, you can do more than that. You can say, okay, what would be the best panel if we wanted to discriminate animals that have cancer versus that don't have cancer? And this is where really the bioinformaticians go to town, and they spend a lot of time applying very different models. Our pipeline is described here. We tend to do quite a bit of filtering of our features. We don't always want to work with the whole data set of features. That could be tens of thousands of features. So, but we always like to apply a suite of machine learning models, including logistic regression, including random forest, SVM, and so on, just to compare and see what the best results are. So what I'm showing you here, these are the area under the curve plots for different machine learning classifiers for different lifetime stages. Notice that it takes me a long time to talk about this because this is a complex data set. But what you see is that some classifiers, for example, if you look at, again, stage 2, some classifiers perform better than others. Some are not that successful, okay? 0.6 area under the curve, 0.7. They're not that successful. And part of that is that we're only using the molecules that we are able to annotate. So there could be better information in the metabolome of molecules that we haven't annotated, but this is the first pass. I'll show you a little bit more about what we do. So what you see here is, for example, that a voting classifier that uses kind of like a consensus approach has the best success for lifetime stage 3. And then you can look at the different families of the lipids, sphingomyelins, ceramides. That helps you map all of these back into lipid pathways. One of the issues with lipid pathways is that the biochemistry is lagging behind. And so we don't know as much as we would need about lipid pathways. So we know what lipids are changing, but we don't always know their biological function. Okay, and that is very frustrating sometimes because we have more detail about the molecules that are changing, but we don't understand what that really means many times. So, of course, working with animal models is nice because you can do very controlled experiments, but we are interested in ovarian cancer in humans. The challenge of doing that is that to be able to do careful studies in humans, in principle, you would need to design cohorts to collect samples under controlled conditions. And that is incredibly costly and not always possible. So many times you're basically studying the samples that you can get. And in the case of ovarian cancer, it's particularly challenging because, like I told you, ovarian cancer is relatively rare. So how are you gonna find biomarkers of early stage ovarian cancer if you cannot detect early stage ovarian cancer to begin with? Those early stage cases are actually found by chance. A patient that goes and has surgery for another reason, and they detect that they had early stage ovarian cancer. So they're incredibly rare. It's very, very difficult. And finding samples that are collected under the same conditions is very, very difficult. But this, I'm going to show you results. These are not published. This is from a very unique data set. And I'm saying that it's unique because notice the number of early stage samples. In this case, early stage human patients is 93. So that's actually quite a good number. If you survey the literature, you're going to see the numbers are typically pretty smaller. And the total number of patients, it's in the order of 300, roughly speaking. I should clarify that when we talk about ovarian cancer is not a single type of cancer. The mice models represent the most deadly type, which is called, it's a high-grade serous carcinoma cancer. But in humans, you're going to see several types of cancers. You're going to see high-grade serous cancers, but also you're going to see things like clear cells. And that diversity in the type of cancer plays against you when you're applying machine learning, because not all these cancers are the same. So you're basically bundling everything into one class. But when you try to stratify and make subclasses, then the numbers are so small that the models are not robust enough. So that is one of the challenges. In this case, we applied a very similar pipeline, actually almost identical to what we did with the animals, because one of our goals is to compare animals, the changes observed in animals with the changes observed in humans. But we cannot compare healthy humans against patients with ovarian cancer. And that's because the way that the samples are collected for healthy humans in this cohort was immediately after anesthesia, and anesthesia affects the metabolome. So you cannot compare healthy patients that are not anesthetized with patients that were anesthetized without seeing a major effect of the anesthesia. So instead, what I'm going to show you is a comparison between patients that have ovarian cancer against patients that had other ovarian conditions that were not cancer, or other types of cancers, for example, cancer of the uterus. Okay, these are other types of gynecological conditions that are not ovarian cancer. Okay, so that's a much more difficult question because the differences are going to be smaller. So this was the first analysis that we did. We did it just based on the lipid families, just the first pass. And what you see here is that for early stage patients, again, most of the patients had the changes were in the phosphatidylcholine, which maps to what we saw in mice. And something I didn't tell you before, but also maps to mice, we see changes in ether-linked phosphatidylcholine, which is also what we saw in animals. And that is reassuring because it connects, it basically validates a little bit the animal model that we're seeing similar things. But of course, there's many other changes in many other lipid conditions. You see here the volcano plots that show the fold change of the different features and their respective p-values. So you see that there's very significant changes pretty much in the whole lipidome, okay, for all the different families, which is interesting, but would that be enough for a diagnostic? This is unsupervised analysis. So what you're looking at here is a simple PCA score plot. And we're looking at the gray are the ovarian cancer patients. The other ones are other malignancies. And you see that there's not a very good separation, as you would expect in a PCA unsupervised analysis, because this is a relatively minor effect size. Of course, as you go to an advanced stage patients, the difference is increased because these are patients that are very sick. So at an advanced stage is very, very clear to map. If you try to find what is the best, absolute best lipid panel to discriminate between ovarian cancer and non-ovarian cancer conditions, you see again that it is possible. So let's see, this is the results for the test set only on early stage samples. So you see that the best that we can get is about a 0.76. 0.76, that's for all cancer types. If you only restrict it to the serous carcinomas, that's almost 0.8. So it's a little bit better, but it's not 0.9. Okay, it's not fantastic. And part of this is that we're doing the analysis in blood, and blood is reflecting the whole organism change. So we're not really sampling close to the ovaries, right? So this may not be the best choice in terms of biomarker discovery. So what else can we do? Well, you can look at the site of cancer. So I showed you the pictures of the ovaries and the tumors and the reproductive system of these mice. One of the things that you can do, this is a difficult experiment to do, is you can basically put all that tissue in a block of gelatin and then slice it and make 10 micron slices and then perform mass spec imaging experiments of those slices. That is not trivial to do, and it takes a lot of practice. And this is work by one of my postdocs, Shin Ma. He's incredibly good at doing these experiments. And what you see here is the outline that basically the photos that I showed you before reproduce this. You see, these are the two tumors. This is for 2 DKO animals at late stage. And this is a TKO animal at late stage, and this is a control animal. And what you see here is ion-specific maps for different lipids. And you see how many of these lipids actually are increased in both DKO and TKO mice, but not in controls. Okay, so some are, but not all of them. The numbers that you see here, this is just for the mass spectrometry crowd. That's just the mass error that we use to annotate, putatively annotate these lipids. Okay, but what this is telling you is that there's definitely lipid remodeling at the tissue level, as you would expect. So how, but that signal effectively gets diluted in blood, right? Because that signal, if these are released in blood, then you lose it. But still, what you can do is you can map all these alterations into pathways. And here you see a snapshot of lipid metabolism and some of the lipid families that we see remodeling, like phosphatidic acids, PGs, PCs, PEs, and so on. And of course, ceramides and sphingomyelins, those are some of the families that are really changing in late stages pretty dramatically. Okay, so you see that some of these changes are quite dramatic when you look at the tissue directly. By the way, these data sets are quite massive and they're difficult to work with because each image effectively is an XY image, but each pixel has thousands of data points. So it's similar to a hyperspectral image. You can perform similar, supervised and unsupervised analysis of these data sets. This is just for a single lipid. This is a phosphatidic acid 32-0. 32-0 means that it has 32 carbons and no insaturations. And you see that in this case, that is a very distinctive marker of cancer. But not all of them are different, like I showed you in the images before. Okay. And then you can do things like PCA, PLSDA, the typical tools that we use in metabolomics to distinguish them. You see here that each set of, for example, here you see that there's 2 sets of red points or symbols. Those are 2 different animals. Okay, so the animal to animal variability is actually quite large. One of the limitations of this experiment is that we cannot sacrifice animals at an early stage because there's no reason. You can't just sacrifice an animal at an early stage. Okay, so it would be nice to see some of these things at an earlier stage than what we're doing. I haven't told you anything about regression. I think I've told you about pretty much every aspect of machine learning. Something that I didn't mention, but it's part of this, is batch effects. Many of these experiments have batch effects, and there are algorithms out there that we use to correct for batch effects in the data set. Particularly, we use one called SURF that it's published in the literature and works very well. But what I haven't told you about is regression. And regression analysis does play a role in metabolomics. I don't have an ovarian cancer example, but let me just show you something else that you may be interested in. So we have a very large center at Georgia Tech that is interested in cell therapies. And you know that stem cells are being used now to treat a lot of diseases that are not treatable with common therapies. But one of the limitations and one of the reasons why these therapies, these cell therapies, are not being approved or they're failing the FDA approval process is because there's no quality metrics in trying to predict their potency, and the mechanism of action many times is not understood. Most of the time is not understood. Okay, so until those hurdles are answered, we're going to be in limbo. So one of the things that we're doing with this center is we're trying to apply lipidomics and metabolomics to understand potency and to understand and therefore the mechanism of action of these cell therapies. So just to briefly show you some results, this is a larger team. So we are the part of the team that we are looking at the intracellular metabolites and lipids by mass spectrometry, but also we work with Art Edison at UGA. He looks at the extracellular metabolites using NMR. And then we work with 2 stem cell specialists at UGA, and they are the ones that basically get us kind of like the ground truth information, the potency information using the assays that are currently accepted as being the gold standard. Those are typically T-cell assays, and there's another assay that is called the IDO assay, and those are the ways that potency is measured right now. So just to quickly show you some results. The good news is that we can definitely build machine learning models to predict potency. This works surprisingly well. We can definitely predict potency by measuring the intra and extracellular metabolites and lipids. And this is just a small selection of lipids, of results. This machine learning model, I wanted to tell you about this. This uses something called functional regression. We are doing that work in collaboration with a company called Evolve Analytics. And this is the different inputs of the different lipids. You see, these are all named lipids, again, because we worked in this case only with lipids that we could annotate, so we can map these back into pathways. But there's a very large amount of information that we're not using here. And I keep telling you that because one of the bottlenecks in the field, how much time do I have, Ivet? 2 hours? 15? Ah, perfect. Okay. So one of the challenges in metabolomics, you've heard me say this almost 7 times already, is annotation of the peaks. Okay. So the difference between proteomics and metabolomics is that in proteomics, you're dealing with biological polymers, that they have a known alphabet. So you have a set of amino acids, and we're talking about combinations of those amino acids and post-translational modifications of those amino acids. But the universe of molecules is contained, if you want to put it that way. When you're talking about metabolites, you're talking about families of compounds that range from lipids to sugars, to amino acids, to nucleobases, and so on and so on and so on. So the molecular diversity is much larger. And when you see a peak in your NMR spectrometer and your mass spectrometer, you don't always know what that peak is. Okay, so you can decide to work only on molecules that are known, or you can work on the whole data set. But this part of the metabolomics pipeline, which is annotating the features, which is basically going from a peak in your spectrum to a structure, that is by far the most challenging part of what we're doing. And it gets very wild. There's quite a bit of advances, but it's not perfect. Just to give you an extent of the challenge, this is a little bit of data from a large consortium that we're part of. This is called the MOTRAPAC consortium, the Molecular Transducers of Physical Activity consortium, and we do all the lipidomics work in that consortium. And we're looking at the effect of physical activity, exercise, in both in animals and in humans. And this is just for animals. This is basically the reason why you need to renew your gym membership. But you see that exercise causes a lot of remodeling in the lipidome, as we all know. But what's really interesting is that the dots in gray, those are unknown lipids, okay, that are changing. And you see that the amount of information, the amount of biological information that we're missing, is pretty huge. Okay, so ideally, if I come back 20 years from now, instead of having a bunch of peaks that we don't know what it is, we know exactly which one of these molecules is. And we can then move on to biology rather than worry about the peaks in the mass spectrum. Okay, so we'll see. So some people they call this the dark matter of the metabolome because it's really like an unexplored space. So how do we identify molecules in this type of metabolomics experiments? Well, there's many ways of doing this. This is just one of the approaches that we use. Let's say that you have, in this case, I'm just showing you one type of cancer that we work with. This is for kidney cancer. We extract the sample with organic solvents, and then you typically do the profiling of the metabolome and the lipidome using LC-MS. I told you that we typically use Orbitrap mass spectrometers because they're very stable, but there are other types of mass spectrometers that provide complementary information. For example, you can do ion mobility experiments to obtain additional information about the molecules. And then you go through your differential analysis, you look at what molecules are changing, you use volcano plots and so on. And then there's all sorts of machine learning tools to understand the data. For example, one tool that is out there that is incredibly powerful is a tool called Sirius. It's a machine learning tool that predicts fragmentation of molecules and therefore tries to give you potential structures for those molecules. It doesn't always work, but it doesn't tell you what the molecule is. It just gives you a long list of potential candidates. One of the things that we've been doing is we've been developing machine learning models to, in addition to predicting fragmentation, to also predict the shape of that unknown molecule. And we do that using a technique called ion mobility spectrometry. And that is basically one additional filter for removing candidates, or if you wish, removing false positives. So if you start with, let's say 600 potential candidates, the addition of all these layers of information helps you get to the most likely structure. So some of you are experts in ion mobility probably, or have worked with ion mobility data. It's a technique that has been around for a very long time. It's the technique that is used to detect explosives at the airport. At least in Atlanta, they always stop me, and you never know what they're going to find. It's also used for biological purposes if you couple an ion mobility separation with a mass spectrometer. And the ion mobility gives you an additional dimension, which is something that we call the collisional cross-section of that molecule. In other words, it's kind of like an averaged, rotationally averaged shape of that molecule. And then you can distinguish molecules if you don't have the right shape that will help you separate. Sometimes it's possible, sometimes it's not, but it does help. So the way it works is like a gas phase electrophoresis experiment, if you wish. So you need to start with ions and you separate them based on shape in the gas phase. So what we do is that we measure collision cross sections experimentally for all the analytes of interest, all the metabolites of lipids of interest. And simultaneously, we build a machine learning model to predict collision cross sections. And the way that we do that is that we codify the structure of the molecules using something like the INCHI keys. And then we calculate molecular descriptors. This is an approach that has been used in drug discovery for decades. So we calculate molecular descriptors for those molecules, and those molecules need to have experimentally measured collision cross sections. And then we build a model, let's say using a supported vector regression model, using some select molecular descriptors. And then we apply that model to unknowns, and we filter those molecules using the unknowns. So this is just, by the way, this is Erin Baker, who used to work here. And this is something that we did with Erin, and it's published very recently, just to show the power and the limitations of this approach. Let's say if you start with a xenobiotic, like this molecule here, and this is the correct match, but you don't know that a priori. This is the experimental value, and what you see here is the window of molecules that you can retain and the ones that you can reject based on a collision cross-section filtering. Okay, so it works, but it doesn't tell you exactly what molecule is. It's one additional filter. If we keep improving the methods, if we keep improving the accuracy of the prediction and the measurement, effectively it's going to become more powerful. So this is just a comparison of some of the algorithms out there. They all work very well. So it depends a little bit on the set of descriptors that they use, how well they do on different families of compounds. The code, by the way, the code for our method is published. It's on GitHub. So if you're interested, I can point you at that. But there's a lot of teams working on this, and it's getting better and better and better. But like I told you before, all of these models that we're developing depend on knowing the identity of the molecule. So, I was showing you all stem cells. You need to know what those molecules are. You can't just put the name of a known peak and try to obtain mechanism of action from that. So it's something that PNNL played a major role in about 20 years ago, and Tom and I were chatting about this earlier, is in understanding how to go from a molecule that is in the condensed phase to a molecule that is in the gas phase, which is necessary to do mass spectrometry and ion mobility and so on. And we tend to forget, the process has been so well studied that we tend to forget that it does have some limitations. But this process is called electrospray ionization, and it's a technique that is used to put molecules in the gas phase without fragmentation in a very soft way. And my lab has very much interested in improving this technique for many, many years. So I want to just show you a few slides about a technique that we call triboelectrospray ionization. So, in electrospray, what you do is you flow your sample through a needle and you create charged droplets, and those droplets shrink and the ions evaporate from those droplets. And then you have a detector typically a mass spectrometer. And this is done with a high voltage power supply to charge the ions. Okay, this is done very, very, very simply. So what we've done is we actually replaced that high voltage power supply with something called a triboelectric nanogenerator. And it's not nano because it's very small, but it generates nanocoulombs of charge. And the way it works is the same way that lightning works. Okay, so when you have rubbing between two surfaces of different materials, you produce charge, you accumulate that charge. And what we do is we route that charge through the mass spectrometer to charge the molecules using triboelectricity. And this has some advantages. So what you see here is you see an electrospray needle, and that is the spray itself. It's pulsed. The reason why it's pulsed, it's because every time so the movie is probably not going to play, but every time we have a robot or linear motor that we move the surfaces back and forth, that produces charge. So this is a pulsed electrospray method, and you can synchronize it with a mass spectrometer if you wish to maximize the signal. So it's very, very sensitive. And the other property of this technique is that it's the electric field is much higher than in standard electrospray. So we're talking about in standard electrospray a voltage of 1,000 to 2,000 volts, and this technique can reach 8,000 to 9,000 volts. So effectively lightning. Okay. So that allows you to do many things. The fact that it's pulsed, it allows you to do what we jokingly call tiny metabolomics. So we want to do metabolomics on very small volumes. Okay, so we've done metabolomics on smaller and smaller and smaller number of stem cells. And this is a paper that came out about 2 years ago showing that you can do metabolomics on stem cells on lower numbers of stem cells. And one thing that we learned when we did this experiment is that the sensitivity was so good that effectively it may be possible to do single cell metabolomics. So we've been asking, well, why would you do single cell metabolomics? Well, if you average across all the cells, you may miss some very unique cells. So if you average metabolome phenotype across a lot of stem cells, well, you may miss some super potent stem cells. And those are really interesting. We want to study those. So that is true for every single cell technique, single cell proteomics, single cell transcriptomics. Sometimes averaging in the batch signal is not what you want to do, but doing single cell measurements is incredibly challenging from the perspective. By the way, I can't get over that little girl, she's so pretty. So doing single cell metabolomics is not routine. And let me just get it out of the way that we are not doing it yet in a successful way, but I'm going to show you what we're trying to do. Okay. So one of the good things about Georgia Tech, it has excellent engineers. So we are working with a couple of engineers with Jonah Temenoff and Craig Forrest groups. Oh, and I don't know if I can play the movie remotely. I don't know if anybody can play the movie for me. There's nobody sitting there. But, oh, it's going to play with the mouse. Perfect. So what we did is we trained the machine learning system. This is recognizing in a microscopy image where the cells are. Okay, that's the first step. The machine learning system moves the microscope, and not the microscope, but the sample plate, and recognizes where the cells are. What you're seeing there are neurons. Okay, this recognizing where the cells are. So the next step is that another robot basically goes down with a micromanipulator and a micropipette tip and pokes the cell. Okay, so that's what you see there. We are using capillaries that have openings that are submicron, typically several hundred nanometers. Okay, and then the robot can basically suck the cytosol of the cell. And then the next step is that this is not automated. We take that capillary and we put it in front of a mass spectrometer. And because the technique uses such a low volume, we're talking about picoliters of sample per spray, then you can get several experiments done on that very small volume. And it's very, very sensitive. So this is what you see here is different pulses of the spray. And what you see here is what the mass spectrum looks like. So big disclaimer, I don't know if this data is correct, honestly. We are doing so many controls yet, okay? But we are working very, very hard. This was done from a single cell, but there's much more work to do. But I just wanted to show you fresh off the benches data, okay? So this is what we're working on right now. But if it turns out that this is true and we can annotate these molecules, then we can start to think about how to do single cell metabolomics and then come back to the computational scientists and be like, how do we analyze these massive data sets that we have right now? So just to finish, I have 2 more slides to tell you about a collaboration that we have here with you guys at PNNL, and it involves also this triboelectric technique to ionize molecules. I used to have a postdoc in the group, Marcos, who's really like kind of like brave guy, and he liked to try new things. And he one day he said, well, look, I did an experiment where I made the electrode, the charging electrode, I made it much, much bigger. And I'm like, how big? He's like about 3 times bigger. I'm like, okay. He's like, well, I have good news and bad news. Typically the bad news means that they broke the instrument. But the interesting thing is that when you increase the size of the electrode, what happens is that in addition to the spray, the electric field is so high that you get a transient electric discharge, effectively a mini lightning, if you wish, between the spray and the inlet of the mass spectrometer. That's a technique that is being used in mass spectrometry for 60 years, and it's called atmospheric pressure chemical ionization. But when you do it in a pulsed way and you do it together with the spray, you get an interesting set of coincidences here. So what happens is that now your lipids, you get a peak for the intact lipid, and then you get another peak for an oxidized version of your lipid. That oxidized version, the oxidation happens in the lipid double bond. And that is very nice because if the lipid double bond is oxidized, then you can break it selectively. And that will tell you where the double bond is. Remember that one of the things that you want to know about a lipid is exactly what the structure is. And positioning the double bonds is not trivial by current techniques. So what IVET is helping us do, which is not trivial, is we normally do the data analysis by hand. That means that Marcos would sit down for weeks and process this data by hand. So now I gave a talk once and Tom said, you know, we have people at PNL can probably help you with that. And I was like, oh yeah, we really need it. So this is the type of data that we're talking about. It has ion mobility data embedded in it, and then it has fragmentation data embedded in it. It's pretty complicated data. And I'm going to save you all the chemistry and all the fragmentation pathways. This is probably more than you need to know, but just believe me when I say that the fragmentations can be very specific and you can detect exactly what type of lipid you are dealing with using this approach. But we're not there in a position to do this, you know, for 1,000 cells at the time, because, you know, the amount of data will be ridiculous, and then we wouldn't know what to do with it. So I'm gonna finish here and allow a little bit time for questions. Just to tell you that the Journal of American Society for Mass Spectrometry, which I'm one of the associate editors, is planning an issue on metabolomics, and that includes computational aspects of metabolomics. So if any of you has anything that could go there, please let us know. You should receive emails soon about this. So thank you very much for the invitation again.
Thank you so much, Professor Fernandez. Thank you so much for this wonderful talk. Impressive all the research. So now we have time for questions. Do we have any questions from the audience?
Thank you. It's great. It's like so many research, so many areas. I'm just wondering, you mentioned a little bit about you're not feeding all the lipids to the models. You will filter them first. And I see your slides saying something like t-test filtering. I'm wondering if that's your like pipeline workflow part of it, or are you using other filtering methods, for example, match to your in-house database or something? I don't know.
Yeah. It's a super good question. And the answer is we use all sorts of different filtering methods. The most common one, when the data set becomes so large, the most common one is we start only with the differential features, and then we remove anything that is highly correlated in that data set. So we're not looking at features that are highly correlated. But you made a very good point, and you said, well, do you filter by matches to the database? And I'll tell you a short story. The answer is yes. But we've had projects, for example, where we were working on cystic fibrosis, and we could predict really well the outcome of certain patients, but then you will look at what the data looked like, and you couldn't really tell what those peaks were. You could say, I can use these peaks to predict the outcome, but we could not identify them. And that is just basically unacceptable, right? Because if you can't explain why it works, it's just, how do you know you're not overfitting your data? You know, you can do all sorts of things, but so we apply a lot of filtering, and filtering has disadvantages. You may be throwing away interesting information. Same thing for the imaging data. We do quite a bit of filtering in the imaging data, and I didn't put any data here about filtering for images, but we tend to work only with features that we do have matches in the databases. We use a program called, I always get the name wrong, Metaspace or Metascope. I always get them wrong. Yes. And so we basically sub-select, but there's many more features that we're not touching. So it has risks. The filtering is a little dangerous. Some data scientists don't want to do filtering. They just want to work with all the data set. And I always ask them twice, do you really want the whole data set?
Do we have any other question? Yes. Please, we encourage the online audience also to post their questions in the chat.
So going back briefly to the analysis of ovarian cancers, this is kind of from outside the biological field, but in astrophysics, the issue of color cuts, developing color cuts for picking out specific stellar objects from a background is very like the issue where you had these very low separation, very low effect size problems. And one of the ways we resolved that in my sort of home field is we applied ML technology to, how do I put this, derive these color cuts for us with a very large data set. And since it seems from the rest of your talk that you do have this extremely large data set, have you considered automating a means of selecting out these early stage markers, or?
Yeah, you're bringing such a good point, and you know, funny you say this, because a good friend of mine who is one of the few people that I know that left a tenure track position to start a machine learning company, and he's a millionaire now. He used to be an associate professor, right? He makes 2 orders of magnitude now the same. He told me once exactly what you just told me. He said, you know, in the field of astrophysics, these problems are being worked on. And that's the extent of our conversation. But if you or anyone in your team is ever interested in looking into the data, more than happy to talk and to share the data. And you know, like, just tell me where to send it. I mean, I think we really owe it to the patients to look at the data the most we can. And the challenge with the human data is that it's not a designed experiment. You know, the doctors collect the patients that show up to the hospital. In the future, I think that to advance the field, we're going to need to basically ask everybody that goes to the hospital, do you want to donate blood to do omics? You know, some countries do it. Some countries do it. Yeah, and they have wonderful data sets. Yeah. But yeah, if you're ever interested, just shoot me an email.